Good morning. This is an audio test for the MPG Primer. This morning's event will begin at 8:30 with Kaitlin Samocha. We will be doing some audio and video testing between 8 and 8:30 this morning, but please stay tuned. The broadcast will begin in about 20 minutes.
This is hers?
So if you want to text me, that's the easiest way to get on it.
When she's going to start? Yeah, that's fine. Text her.
Where do you want me to put this mic?
Right here. Thank you.
Yep. And actually go down it.
Okay, good morning, everyone. We're going to get started with a primer. My name is Kaitlin Samocha, and I'm currently a postdoc with Mark Daly here and at MGH. And it's really exciting to be able to talk to you today about studying de novo variation and studying genes that appear to be intolerant to mutation or constrained against mutations within the human population. So you might be wondering what de novo variation and constraint have to do with each other. The big thing is that they both use this mutational model. So I will be discussing this mutational model in detail later, but this is the basis of both of these. In many ways, you can think of me as round 3 of a number of primers on studying rare variation. So a few weeks ago, we had Ben Neal doing the more classical rare variation approaches. And then just before Thanksgiving, Daniel MacArthur was talking about these large aggregation databases such as ExAC and Nomad, which we're using to study the variation in patterns within the human population. So I'm really using pieces from both of these talks as the basis of my work. I'll remind you of what is relevant for my talk, but these two will also be helpful for you to understand studying rare variation within populations. So kind of the issue with studying rare variation, and Ben went into a lot of detail on this, is that if you try to use the tests that we use on common variation for rare variation, they just fail. So this is a QQ plot for about 2,800 cases with schizophrenia and about 4,300 controls. And here they took a minor allele frequency of 1% and all of the variants that were rarer than that. And this QQ plot shows on the x-axis the expected p-values from these association tests, and on the y the observed values. And you'd expect, if it's null, that they should fall along this diagonal line. However, when you do this testing of rare variants, you see that they're deflated. And the reason is because you're underpowered. These variants just don't have enough observed counts for you to be able to use this type of testing methods that have been discussed before. So Ben went through a lot of different ways of population genetic modeling and how you should start to approach it, but the big take-home point is that you need really, really large sample sizes if you're going to try to do case control analyses of rare variants. So this was his take-home figure from his talk. On the x-axis here is increasing selection. So the further right you are, the more deleterious the phenotype, the disease that's caused by this variant or set of variants is. So over here, this is very, very severe, and this is weaker selection. And on the y-axis is the number of cases you need, assuming infinite number of controls, essentially, in order to find an association. These colored lines are varying effect sizes, but if you kind of make this into a table, what we see is that for weak selection, with an effect size of 20, maybe you only need 260 cases in all of those controls I mentioned. But as you move to stronger and stronger selection, or as you move to variants or classes of variants that have smaller effect sizes, you need really, really large case sample sizes, up to 2.8 million in this corner over here. So really, if you're going to try to do rare variant association studies, you're going to need a lot of cases. So there's some strategies to get around this, and Ben really went through all of it, and it's also discussed in this Zuck et al. 2014 paper. But you can focus only on those mutations that are disrupting, can start to filter your missense variants, so you're including those that appear to be most likely to be contributing. You can use isolated populations like Finland, or you have to start aggregating in other ways. Another strategy that he mentioned but didn't go through was studying de novo variation. So de novo variation is variation that's seen in a child but not seen in their parents. So this is in their somatic genome overall. So here, if dad and mom both have AA at a particular site, the child might have AC. And that C allele, that's our de novo variant overall. So given the fact that DNA has this natural property of mutating all the time, we expect to see about 70 to 100 de novo variants in everyone's genome. Everyone in this room has 70 to 100 de novo variants in their genome. They just happen. Given that number, we expect to see maybe one in exome. Unfortunately, to find these, or not so unfortunately anymore, you have to sequence. So these are not really on chips. This is sequencing 3 individuals to find all of these rare variants. But a pro of studying these incredibly rare, newly arising variants is the fact that they can be more deleterious than the ones that you see at higher allele frequencies in the population. So, for example, this de novo allele here could lead to the complete inability to have children. That means that this would never, ever, ever be passed on. You're not going to see it in 20% of the population, but you can see it de novo. The only thing that's been removed by selection so far with de novo variants is those that are incompatible with life. So given how large the human population is already and how big our genome is, we would expect that if we sequence the entire world, we should see every single de novo variant that does not lead to embryonic lethality. So a con of this is that these de novo variants, again, I said 1 an exome, there's a lot of territory in your exome. And if you're trying to do a gene association test with this, they're just not going to pile up very quickly. How many do you really expect per gene? And that's the major struggle here. So why are we even studying this? This is those super low frequency ones that's going to require these massive numbers of cases. But we know de novo variation is important to disease. So here the child is now affected, and we're sequencing both parents and the affected child. And after the exome sequencing work sort of came into its own, there were a bunch of studies that found de novo variants tied to very severe and likely monogenic, or only a couple of genes contributing to these disorders. So Schnitzel-Gideon syndrome, for example, they found de novo missense variants in 10 unrelated people in this one particular gene. Kabuki syndrome is very similar. Of their 10 unrelated individuals in their initial cohort, 8 of those 10 had a de novo protein truncating variant in the exact same gene. And this is incredibly unlikely to happen. Again, if you're expecting 1 per exome, and you take all of these cases and they're all hitting this exact same gene, and they're the same type of change, not the exact same change, I should note, that's extremely unlikely to happen unless these types of variants are actually associated with the disorder. So this, in both of these cases also, they had a number of other patients that had the same types of mutations in these genes, but they couldn't confirm that they were de novo because they didn't have parental DNA. So for this talk, I'm going to be focusing more on the autism spectrum disorders. Now, this early work, these were really severe, and it seemed to be a little more straightforward genetically. When we move into the autism spectrum disorder space, as you'll hear a lot more from Mike Tukowski's group later today, if you stick around for MPG, is that the autism spectrum disorders appear to be more complex in terms of their genetic architecture. So as a very brief definition of the phenotype, this is a collection of severe early onset neurodevelopmental disorders that are characterized by impaired communication, particularly verbal communication. These patients often have repetitive or stereotyped behaviors. So our group and others, using exome sequencing technology, wanted to begin to understand the genetic basis of the autism spectrum disorders. So there had been success studying de novo, so these newly arising C and V, so these massive copy number variants that duplicated a large number of genes or deleted a large number of genes. When you do exome sequencing and you can find these de novo smaller changes, single nucleotide changes or indels, it points to a specific gene. So these are a little easier to interpret biologically. So in 2012, 4 papers came out, and across those papers, roughly 945 trios were sequenced, so mom, dad, child, and 350 or so unaffected siblings of the autism siblings, which you can view as controls. But across all of these autism cases, there were 35 genes that had multiple missense or protein truncating variants. So it's not like one gene rose to the top. Unlike those other disorders, we didn't find this whopping signal. And in fact, even though we sequenced nearly 1,000 autism individuals and their parents, 5 genes only had 2 de novo protein truncating variants. None of them even had 3. So this is not like what we saw before with Kabuki syndrome, where you had 8 out of 10. This is, 2? That's pretty bad. And even if we found 0 in controls, a 2 to 0 is extremely extremely hard to make look significant. You'd need thousands and thousands and thousands of controls, but it's really hard to convince people to sequence controls to look for de novo variants, because that's sequencing 3 people and one of them doesn't have a disease. I ideally would like more of these, but trust me, it's hard to convince people to do that. So if you're going to use multiple de novo variants in a gene to indicate that that gene is associated to disease, we're going to have to start sequencing more people. We didn't get the pileups that we were hoping to see. But as you sequence more people, given this background rate of mutation, you're going to see multiple de novo mutations in a gene just by chance. So if we sequence 10,000 control trios, so dad, mom, and the child doesn't have autism, Titan, for example, is definitely going to have multiple de novo variants in it. Titan's a little unfair, I'll say that. It's the biggest gene, it's massive. But plenty of other genes that are much smaller will also start to have pileups. So we need to be able to figure out when we're seeing multiple de novo variants in a gene, is that interesting because it's related to biology, or is that just happening by chance because there is this background rate of mutation? So we really need to have an expectation here. So when we move into our studies of de novo variation, we can evaluate any kind of interesting findings. And in order to establish that expectation, we made a model of mutation. So this is the key thing for both the studies of de novo variation and constraint that I'll get into later. So our model of mutation starts with intergenic SNPs from the 1,000 Genomes Project. We chose intergenic SNPs because they should be relatively free of severe selective pressure. And we focused in on regions, intergenic regions again, that were orthologous to chimp. So we have an idea which allele is ancestral, which is the older one, and which is derived. So which way did that mutation move? So let's say we know, for example, this SNP here it used to be a G and then it moved to an A at some point during the human lineage. So now we want to know the probability that we would see that type of mutation. It had been established before we were doing our work that the best way to determine the mutability of a single base is to look one base to the left and one base to the right, so to look at this trinucleotide context. So we pulled that out for all of these intergenic SNPs and used it to create a mutation rate table. So for every trinucleotide mutating to every other trinucleotide, we have a probability of that occurring. Again, it's only this middle base that's changing here. Now, since this is based on human data, we can apply it to any stretch of human DNA. But at the time, and for the most part still now, people are doing exome sequencing. They're only sequencing the coding regions and not the whole genomes. So we applied this mutation rate table to all of the coding base pairs in the human exome, as well as the essential site bases that are on either end of exons. And so for each of these bases, we determined the probability of mutating to the 3 other possible bases and then predicted the change that would have in the protein product, for example, synonymous, missense, etc. So we get, at the end of the day, is a per gene probability of mutation that you can break up by different mutational classes. Now this is a very simple model on its own. It only really takes into account the length of the gene, since we're looking at every single one of these bases, and it takes into account local sequence context. We did make 2 adjustments on top of this. The most important, of course, being sequencing depth of coverage. So if you don't sequence a gene at all, we're not going to see any de novo variants there. We just don't have any reads to call it. So we don't want to expect to see any de novo variants there. This is also really critical for our work later on, but sequencing depth is the biggest thing that we correct for. Secondarily, we correct for the fact that there are these large, megabase scale differences in the mutation rate between humans and other primates, so humans and macaques. And so we make a very modest adjustment for the local large effect mutation rate in all of these scores. So now we have per gene probabilities of mutation that take into account sequence context as well as depth of coverage, and we can use them to analyze some of our de novo variant studies. So ever since 2012, a lot of work has been done into sequencing families where the child has an autism spectrum disorder. And at the end of 2014, 2 papers were released from the Autism Sequencing Consortium and from the Simon Simplex collection that altogether had about 4,000 cases with autism and about 2,000 of these unaffected siblings. So again, these are not true controls, as in they're not from a totally different family, but they do not have autism themselves. So some of the key questions we can have when we're studying de novo variation in these cases is, are we seeing more deleterious variants in our cases than we would expect to see? But most importantly, are there any particular genes that we can highlight because of their burden of these de novo variants? And failing that, potentially we can highlight pathways. And all of this is in a way to associate disease genes or disease pathways with the disease itself. We're trying to pull out the relevant information. So to answer the first question, I'm showing you here is the observed number of de novo events per exome compared to the expected number of de novo events per exome for the 2,000 or so unaffected siblings. So this is broken down by synonymous missense and protein truncating variants. We can see is that missense, let's say we expect about 0.6 de novo events per exome, and we expect about 0.62, and the unaffected individuals only see about 0.6. So we're matching fairly closely. But when we look at the individuals with autism, we see significantly more de novo missense and de novo protein truncating variants than we would expect to see. So this enrichment here for the missense variants is not quite as large as you see for the protein truncating variants, but it is significant. So what this indicates here is that there's some signal both in missense and in protein truncating variants. We'd like to be able to figure out if they're highlighting particular genes. So we can do that since these probabilities of mutation that we have are per gene. So when we look at the unaffected individuals, we see that there are a handful of genes in the 2,000 or so unaffected individuals that have 2 protein truncating variants. However, none of them cross the significance threshold that we set at 10 to the minus 6, which conservatively corrects for the number of genes, the number of tests that we're doing. And this is really up here to highlight the point that just because you find multiple de novo variants in a gene, it doesn't mean that it's related to anything. It's just going to happen when you sequence people. This is very different, however, when we look at the autism spectrum disorder cases. Again, we have about 4,000 trios here, and we have a couple of genes that are crossing this really strict significance threshold. For example, CHD8 has 7 de novo protein truncating variants in these 4,000 or so individuals, which is extremely surprising given what we would expect to see. And a lot more work has been done on CHD8. We've found many more de novos in other studies. They just weren't included in those too. So this has really arisen as something from the de novo studies as an important autism disorder gene. And I don't know if Mike will talk about it or not, but this has definitely had a lot more work done on it. When we look at some of the other genes that popped up, SYNGAP1 and ARID1B have significantly more de novo protein truncating variants than we would expect. These have been associated with intellectual disability type phenotypes. And given that a lot of autism cases have lower IQ, that's potentially not surprising to see. But I'd also like to point out here that this is a conservative way of counting things in and not so much a way of counting things out. GRIN2B is another gene that's been associated with intellectual disability, and the 3 de novo protein truncating variants in the autism cases here don't quite make it cross that threshold. But given that it is affiliated with intellectual disability, maybe it could be contributing to disease in some of these patients as well. But this highlights that maybe not all of the genes that are important are going to get lucky enough to be hit as many times as we need for them to cross that significance threshold that we have. So possibly we can just group things together by pathways or by networks that we know that we hypothesize might be important. And we can use the probabilities of mutation again to evaluate if we're seeing burden on this gene set. So you can use any gene set of interest and determine the expected number of de novo variants that would fall onto that. So let's say, for example, the gene set you're looking at represents 10% of the mutability of the exome. That means that 10% of your de novo variants, synonymous missense, protein truncating, 10% of all of those should fall onto this gene set just by chance. So if we see 12%, maybe that's not surprising, 30% would be. So the gene set that a lot of people were excited about when we were first doing these autism sequencing studies were the targets of the Fragile X mental retardation protein, or FMRP. This is what's lost that leads to Fragile X syndrome. And Fragile X syndrome, while mostly, again, intellectual disability phenotype, a number of these patients do have autistic-like features. So this is kind of the connection between autism and FMRP. And FMRP is a really critical neuronal RNA binding protein. So a ChIP-seq experiment was done and found about 850 targets of FMRP, and that's the gene set that we're testing for enrichment. So what I'm showing you here is the fraction of these FMRP targets that have a de novo variant split up by synonymous missense and protein truncating. The dashed line indicates what we'd expect. So you can see this does actually represent about 10% of the mutability of the exome. And the green bars are for the unaffected individuals. So the unaffected individuals, their distribution of de novo variants doesn't look any different from expectation, or at least not significantly so. But when we look at the patients with an autism spectrum disorder, we are seeing significant, but again, modest enrichment in de novo missense variants for the targets of FMRP, but a very large enrichment when we're looking at the de novo protein truncating variants. So this excess of de novo variants that we were seeing in the autism spectrum disorder patients is falling into particular genes and into particular pathways that appear to be relevant to disease. Yeah.
Just, you pointed to the very high bar of 10 to the minus 8, but you people have now had enough where you've seen several 10 to the minus 7, 10 to the minus 6 in early studies, and then with larger n's. How do those break out? Do the majority of those actually increase over 10 to the minus 8? I understand if you have a pathway to sort of pick them out, but if you're just picking out new candidate genes, how do they break with increasing numbers?
So I'm being asked, as you sequence, so this is 4,000 trios, but as you continue to scale up, the question is, how are we seeing continued pileups in the exact same genes, or are we seeing it spread out more? So it depends on the phenotype. For autism, we are seeing that a few more candidates are rising to the top. Some of these genes that I highlighted, the 7 or so that significantly crossed that threshold back here, a few of these had multiple variants in the earlier studies when we only had 1,000 or so. CHD8 continues to just have things piled on. A couple more like SCN2A, ARID1B, SYNGAP1, they've seen some pile up as well, but there are a lot of genes that are getting multiple hits. Now, that doesn't mean that everything did. Our first studies, there's a gene called CATNAL2 that had two de novo protein truncating variants in about 1,000 cases, which looked kind of interesting, but it has never had another de novo protein truncating variant in autism. So it's a mixed bag. In intellectual disability and developmental delay space, they get even more pileups. The Deciphering Developmental Disorders paper that was put on the bioRxiv in June, they have in about 4,000 trios some genes with like 30 de novo protein truncating variants in 4,000 cases. So I think it partially gets after the genetic architecture of what you're looking at. We'll skip through this. Right. Go ahead.
You probably may come back to this later.
Yes.
This kind of very modest missense signal, were you able to refine that later with some, are you able to refine this even better?
Yes. So I won't get as much of a chance to talk about it, but a lot of the work that we've been trying to do is to find ways to highlight subsets of missense variants, de novo missense variants that we think are more likely to be contributing. It has not yet been fully refined in the same way, but we've actually even been refining this protein truncating signal as well. So we're working on it. It's not quite there. Right. So studying de novo variation in these individuals with an autism spectrum disorder was highlighting what appear to be biologically relevant genes. However, we can also study patterns of variation, shifting gears, if you will. We'll also be studying patterns of variation in healthier populations. And this can aid in variant prioritization. So ever since we've been sequencing large reference populations. So back in the day, we had the 1,000 Genomes Project with about 2,500 individuals. The NHLBI's exome sequencing project came out 6-7 years ago with 6,500, and then a few years ago, Daniel MacArthur's group put a lot of effort into corralling a bunch of data and jointly processing it, which they released as ExAC, or the Exome Aggregation Consortium data. So this was about an order of magnitude larger than anything that was previously publicly available. I'll underline that. A lot of sequencing has been done, but publicly available is the key thing here. And it was much more diverse than the exome sequencing project. So pros to this is that we got rid of individuals that were related, and we dropped samples that couldn't be publicly shared, and we removed individuals with a known severe pediatric disease. So these represent a reference population. They're not super controls. They're not perfectly healthy. Some of them have diabetes. Some of them have high blood pressure, but no one is a perfect control. So this is the best type of look into a general population that we can get after. Now, if you saw Daniel's talk a few weeks ago, you'll know that we also just kind of outdid ourselves by releasing at ASHG a sample set called NOMAD for the Genome Aggregation Database, which has twice as many exomes and now about 15,000 genomes. Since we are still working out some of the kinks in this data set and it's brand new, all of the analyses I'm talking about, when I say ExAC, I mean only the 61,000 or so individuals in that original release. So ExAC was already proved to be incredibly useful within a clinical context. For example, if you're trying to use a minor allele frequency filter in terms of studying rare disease patients, you see is that ExAC drops the number of candidate loci that you need to dig into a little more by about 5-fold. So here is the number of variants that remain after you apply an allele frequency filter about 0.1%. So if you use the ESP data set, again, 6,500 individuals, you're left with hundreds of variants, and these are for the different populations. But when you use ExAC, it drops down below 200 for all of these different ones. So I encourage you, if you're curious about this, to either read the ExAC paper or to listen to Daniel MacArthur's MPG Primer from a few weeks ago, because he does step through it. ExAC also is able to allow us to identify pathogenic variants that were hanging out in some of our disease-associated disease variant databases that are just way too frequent to actually be pathogenic. Again, won't be talking about that as much. So ExAC's been useful in variant interpretation, but we also know that it can be useful highlighting genes themselves, not just variants, that appear to be disease relevant. And the reason this is so important to do is that when you're sequencing people, you're finding thousands upon thousands of variants, and you need some way to be able to prioritize those that appear to be more likely to be contributing to that person's disorder. So there's a large number of variant level pieces of information you can take into account. For example, only focusing on protein truncating or using the large number of in silico tools that predict the deleteriousness of missense variants. You can also use whether or not it's been seen in the ExAC database. If you have a patient come in and you think you found a really severe, fully penetrant, if you believe that it exists, from talking to people earlier, fully penetrant allele that leads to this really bad disease, and you look it up in ExAC and you see it in a couple thousand people, that's not the variant you're looking for. So this is where that's been useful. There are also gene-level pieces of information. So if you have a disrupting variant in a gene that's already been associated to disease or is expressed in the relevant tissue, you believe, that's another way to prioritize. But our group and others have been trying to pull out genes that appear to be intolerant of mutational changes just overall. So we call these constraint. And to walk you through the logic of this, I'm showing a gene that's relatively tolerant of mutational change on the left and one that is intolerant or constrained on the right. So again, getting back to that natural property of DNA where it's just going to mutate, we're going to see different alleles arise in individuals in this population. And in a tolerant gene, maybe one of these alleles leads to some deleterious phenotype and therefore is removed from the population by selection. But in a constrained gene, and that's really sensitive to all of these changes, many of the alleles that arise in this gene lead to this deleterious phenotype, and therefore they're removed from the population. So what we see over time in grouping individuals together is that a tolerant gene will show a number of variants in between individuals, where a constrained gene will be depleted for the expected amount of variation. So we're looking for a signal like this, and this is one of those signals that we use to indicate that these genes are intolerant of variants overall. They'll note that we do have to have an expectation of how much we expect to see. And so we were able to use our mutational model on the 6,500 individuals in the NHLBI's exome sequencing project to identify a set of genes that we thought were intolerant to mutation or constrained. Now, David Goldstein's group also used that same data set in an orthogonal metric, and they agreed with us on which genes appeared to be intolerant. And what we both found was even with a smaller data set, we were able to highlight genes that were depleted of variation, and they were enriched for known disease genes, and we found piling up of de novo variants in patients with autism spectrum disorders and intellectual disability and epileptic encephalopathies. Those all piled up in the genes that we highlighted, indicating that not only are these known disease-relevant genes, but they do appear to also be medically relevant in the de novo space. But this was a couple of years ago. Now that we have ExAC, we wanted to double check that our mutational model was still doing a good job of predicting the number of variants. So here I'm showing you in the bottom left corner the correlation between the observed number of rare synonymous variants, rare being a amino allele frequency less than 0.1%, and the number that we predicted from our model. So every one of these little dots, if you can see them, is a gene, and observed is on Y, expected on X, and we see a very tight clustering along the observed expected line, which matches with our R-squared of 0.96. So we are doing, with this relatively simple model, a very good job of predicting synonymous variants. Now, since these are relatively free of selective pressure, this was a nice check. When we look at missense and protein truncating variants, however, we begin to see that this cloud falls off that observed expected line. In particular for protein truncating variants, we're seeing far fewer protein truncating variants across the board than we would expect to see. And this is because protein truncating variants as a class are more deleterious than synonymous variants, again, as a class. So our mutational model is selection neutral. It just says this is how much you should see things popping up. It doesn't deal with any kind of selective pressures at all. So we would expect to see that the protein truncating variants don't match our expectation from this model. So we were able to do these types of analyses to find genes that appear to be missense intolerant. But one of the things that the ExAC database allowed us to do that was incredibly hard, if not impossible to do before with the smaller data sets, was to evaluate the constraint against protein truncating variants. And the reason this is so challenging is because there aren't that many opportunities in any given gene to make a protein truncating variant, if you're thinking of essential splice site disrupting or nonsense mutations, for example. I will also touch upon a little later, trying to refine the signal of missense constraint to regions within genes, but these are analyses that really required a much larger data set than when we were working with before. So focusing on that first point, what we want to be able to do is highlight genes that are extremely, extremely intolerant of losing one of their copies. So these are intolerant of these loss of function variants. And note, I've shifted from protein truncating to loss of function because loss of function here actually indicates some idea about mechanism. So we assume protein truncating variants lead to a loss of function, but that doesn't always happen. So just kind of stick with it. None of you looked super lost, so I'll go with that. Right, so if something's intolerant, extremely intolerant of loss of function variation, we shouldn't see that many within reference populations like ExAC. The mechanism here would be haploinsufficiency. So haploinsufficiency means you need both copies of a gene to be working to maintain the normal phenotype. If you lose even one copy because of a protein truncating variant or because of a loss of function variant, then you get a disease or an altered state. So I can't promise that these are all haploinsufficient, but that appears to be the way that it's working. So we created PLI, or the probability of being loss of function intolerant, to be able to identify a set of genes that we were fairly confident, due to its loss of function depletion, fairly confident are intolerant of loss of function variation. The logic behind PLI is that we assume that there are three natural classes in terms of their ability to tolerate loss of function variation. So there are those genes that can tolerate completely being knocked out, losing both copies, homozygous loss of functions. There are other genes where you can lose one copy, but you can't lose both. So they're tolerant of a heterozygous loss of function, but not a homozygous one. And this final class here is intolerant of even losing one copy. So it's intolerant of a heterozygous loss of function. So you can, yeah.
So for this definition, though, by intolerant, that means you don't get to adulthood without being recognized.
Yeah, that's actually a point I'm going to make a little later, but these are extremely deleterious. It's not necessarily you don't make it to adulthood. It could just, you could make it to adulthood and not have children, and that's also going to have a pretty bad effect.
Well, the reason that I raise it is because, if I recall correctly, if you look at the Ldl receptor, which causes familial hypercholesterolemia in a heterozygous state, it doesn't pop up on this. So I guess because there are a lot of people with familial hypercholesterolemia that were getting sequenced, or because that although it's not a good mutation to have,
Okay.
people still get to adulthood.
Yeah, so what I'm being asked about is just trying to reinforce whether or not this is only getting after early onset, and I'd say it's mostly getting after early onset in terms of the Ldl receptor and hypercholesterolemia. So there are a number of caveats to why there are some genes we would expect should be more depleted but aren't depleted. I didn't put those slides in this presentation because that's getting into really deep cuts. I'm happy to talk to you about that a little later. But we are pulling out very severe, you don't have children or you don't make it to adulthood types of conditions generally. Other questions? Good? Yeah.
Isn't this just a reflection of reproductive fitness per se, and not really a definition of whether something lasts to adulthood? Because, this is pretty much a reproductive fitness question.
No, and getting into this database, you have to have not had a disease as a child. That would exclude you.
Again, there might be a few people that slip in, but it's known severe pediatric. If they had something severe, they're probably not being included as a control in other cases, but I have to throw a lot of things like probably in there, right? So you can think of these gene sets as like a null, recessive, and haploinsufficient. So null genes are like olfactory receptors. In the human lineage, it looks like we can mutate them this, that, and every other way. It doesn't appear to have any apparent negative phenotypic consequences. For this middle set, again, they can lose one copy but not both. This is like recessive disease genes. When we look at a set of manually curated recessive disease genes, we see that they have about 46% of their expected loss of function variance. This final set, again, is what we're interested in, those genes that look haploinsufficient. And so a set of established haploinsufficient disease gene have about 9% of expectation. So PLI is really the probability that a gene falls into that last category. It's the probability that its loss of function depletion looks most like it's haploinsufficient. So PLI and our other constraint metrics are up on the ExAC browser. If you go to the genes main page, they're up in the top right corner. We also have them for download on the ExAC FTP site. But when we're talking about PLI, this is the distribution that we see for all genes. And it's really bimodal, and we've made it that way. We wanted PLI to be a yes-no. This does not get after how severe the selective pressure against something is. It's a yes-no, does it appear to be intolerant. So we drew a line at PLI of 0.9 and said everything above that, this is the set that appears to be likely loss of function intolerant. And across all genes, it's about 17 to 18% of them that fall in this high PLI tail. So you wanted to throw some gene sets at that. We see, thankfully, is that these established haploinsufficient disease genes are enriched in the high PLI tail. And interestingly, it even stratifies with the severity of the disease. So disease genes that cause a more mild phenotype, but in a haploinsufficient manner, so you lose one copy and you get the phenotype, are not as enriched as those that cause more severe early onset, like death in childhood types of phenotypes. So dominant disease genes are also enriched in the high PLI tail, recessive disease genes are depleted, and another control, olfactory receptors, again, those things you can mutate all the way, those are completely absent. If we saw them in this high PLI tail, we were doing something wrong, so it's good they're not there. So just one thing I'd like to note on this slide is that of the 3,200 genes or so in this high PLI tail, only 21% of them have a disease-associated variant in ClinVar. If we use HGMD or OMIM and we start to bring this list up, it rises to maybe 27 or 28%. So that means that the vast majority of these genes that are massively depleted of loss of function variation in the ExAC database have no associated human phenotype. Not all of them will cause disease, but certainly other ones in there will. Possibly this is where embryonic lethals are hanging out. We don't exactly have a very good understanding of embryonic lethality in humans. We've been doing much better in the mouse space on that. We'll definitely find more disease-associated genes in here where we just haven't quite tied disease and disruptions in the gene together. But as was kind of asked before, what is PLI truly capturing? And here it is severe. So if something causes a skin phenotype, but you can still grow to adulthood and have children, or have most of the children you'd be expected to have, then you're not, even if you're haploinsufficient, you're not quite as enriched as something that, again, leads to cardiac arrest at age 7. They work in a dominant or haploinsufficient manner. So these recessive disease genes, again, we know they're important to human disease. Some of them lead to very severe phenotypes, but they don't appear to be as depleted as the haploinsufficient disease genes are. They can tolerate some loss of function variation in the population. And finally, yes, early onset is important. BRCA2, which we know is important for disease, definitely associated with breast cancer and ovarian cancer, has 52% of its expected loss of function variation in the ExAC database, which gives it a PLI of 0. This, again, is a human disease gene, but it strikes after the reproductive age for the most part. So this is a case where it doesn't have a really high PLI score. Just a few notes about the genes that we have. High PLI genes tend to be the center of hubs, so they have a lot of protein-protein interactions, more so than ones that are more loss of function intolerant. The genes with the highest, or the gene sets with the highest median PLI are really core biological processes like the spliceosome and the ribosome. And these are genes that are expressed at higher levels and in more tissues than more loss of function intolerant genes. So this is really a set of important genes that you need to have around. So there's a lot more of research that has been done in kind of the PLI space, but I want to make a few notes about the missense space. So loss of function and protein truncating variants we assume to be the property of a gene for the most part. It doesn't matter if it hits at position 10 or position 50, it should have roughly the same effect. Of course, there are caveats to that. However, missense variants we know can have very different effects depending on where they fall in the gene and the particular amino acid substitution. So I'm not going to go into as much detail about this as I could, but I'm going to highlight how we've been using ExAC to find regions within genes that appear to be missense constrained. So for example, if you have a gene here showing you the observed number of missense variants over the expected number of missense variants for each of these exons, if across this gene it has about 50% of expectation, the question is whether that 50% of depletion is uniform along the gene or whether there are regional differences. And so if you're looking at this, you see already this first exon has most of its variation, this last exon is really depleted. So what our method might do is split it and say these first two exons appear to be unconstrained because they have the vast majority of their expected missense variation, but these last two exons are very depleted. So this is the constrained region, and this section is driving the overall gene depletion. So when we apply this to genes, we chop some of them up, 15% or so, we chop them up into pieces and have found regions that are severely missense depleted. And so in this graph, what I'm showing you is by their observed or expected missense variance breakdown, the number of coding base pairs. So here in this pink, this means it has 60% or more of its expected missense variation, and that's really the vast majority of the exome. We get down to very, very small numbers when we're talking about depletion below 60% of expectation. So remembering these de novo variants we talked about before, when we looked at the de novo variants that come from control individuals, or those unaffected siblings, we see that they fall into regions as we'd expect by the breakdown of base pairs. So they're falling just based on the target size. By contrast, when we look at established pathogenic variants in these severe haploinsufficient disease genes, they are incredibly enriched in this most depleted 14% or so of the coding region. So this is kind of what we'd expect, 14%, and we're seeing over 80%. This is a massive enrichment. Now, when we begin to look at de novo variants from our cases with a neurodevelopmental disorder like autism or intellectual disability, we see that they fall in between the two. So this is how we're starting to try to refine the signal. Again, we know that a lot of the de novo variants that we identify in patients are going to be benign. They're going to be this background rate, but we're going to be able to start to use where the variants themselves fall, in particular within the genes for missense variants, to highlight those that are falling into these most missense depleted regions. And just to give you a kind of quick example, and I know Daniel showed this one, this is a severe haploinsufficient disease gene, CDKL5, causes a number of fairly with seizures, intellectual disability, a number of pretty bad phenotypes. And when you look at the gene itself, all of these stars are where there are disease-associated variants. And this is where we highlight the gene as constrained. So we didn't have this information. We were totally blind to that. So just using patterns of variation in relatively healthy individuals has allowed us to highlight exactly where pathogenic variants are piling up. Now again, you could have just looked at this picture and figured that out, but there are more subtle cases where this will become more important. So just in the last couple of minutes, I'm going to talk to you about a story where we've been able to use the ExAC database and information about constraint to interpret de novo variation. And these de novo variants I'm going to be talking about again come from our roughly 4,000 cases with an autism spectrum disorder and 2,000 unaffected siblings. And now we're adding cases with intellectual disability and developmental delay. So with all of these de novo variants, what Jack Koshmitsky, a graduate student in Mark's lab, did is he just recapitulated that we see a much higher rate of de novo protein truncating variants in cases with autism, that's this purple bar, and cases with intellectual disability or developmental delay in this blue bar compared to the unaffected siblings. So now we're comparing to the unaffecteds and not to an expectation. So 1.5, 2 times higher. This is for everything. But what Jack found is that he took every single de novo variant and asked whether it had been seen in an individual in ExAC. So just because it's de novo doesn't mean that it hasn't happened before in the human population. In fact, de novo variants can arise at the sites of fairly common variants in a particular family. What he found was all of the signal is found in those de novo protein truncating variants that weren't seen in anyone in ExAC. So all of the de novo protein truncating variants, again, these should be the most severe types of variants. If they were seen in an individual in ExAC, there's no difference in the rate in between cases and controls. All of that happens to fall into those de novo protein truncating variants that were not seen in ExAC. And it's not something magical. I mean, if you look at the de novo synonymous variants, it doesn't matter whether you're looking at all of them, those that were in ExAC or those that weren't in ExAC. There's no significant difference between the rates at all. So this is really specific to these protein truncating variants. Yep.
Can you go back to
Yes.
So even the unaffected siblings have a high frequency of de novo mutations that are not in ExAC.
Yes. So overall, about 30% to a third of all de novo variants are seen in ExAC. Given the 61,000 individuals, if you look at autism spectrum disorders or schizophrenia or congenital heart disease or or controls, about a third are seen in the ExAC database. So so this is just that representation of about like a third, 2/3 or so.
Is the idea that 2/3 of all de novo mutations are bad for you?
No, not at all. Not at all. This is just a way to try to highlight the signal. So it's actually more like the opposite. If you have seen it in someone else, it's less likely to be the variants that you want. It's less likely to be a variant that is contributing to risk because it's been seen in other individuals. Again, this is sort of assuming severe phenotypes like autism or intellectual disability. This might not be the case for other disorders where de novo variation isn't contributing as much. But yeah, not everything in even the unaffected siblings is seen in other people. Like they'll have some things that arise.
I was struck by the same thing, and I wondered, isn't that a subclinical oligogenicity issue? In other words, that some of these variants that the unaffected SIBs are carrying represent a first hit that doesn't affect them, and the sibling that is affected now has a second hit.
So the question is whether this represents some oligogenicity in the data set and that the unaffected individuals might be carrying potentially 1 hit, but they need a second hit to actually get disease. I'm not sure I'd say that. I mean, everyone's going to have de novo variants. Potentially, some of these could be contributing to a phenotype, but I wouldn't say that. It's just.
Then how do you account for the enrichment?
This isn't an enrichment at all. This is. It's not. There's no enrichment here. This is just saying the rate that you see, the rate of de novo variants in unaffected siblings drops from about 0.09, which is what we'd expect, and when you split it out by whether it's been seen in ExAC or not, it splits about 1/3, 2/3. Yeah. Right. So using the ExAC database allowed Jack to highlight a subset of de novo variants. Again, those that aren't in ExAC that appear to be the ones that are containing this burden that you see in cases. And even more particularly, this only happens when those de novo protein truncating variants are disrupting one of these high PLI genes. Okay, so again, Here I'm showing you on the left all of the de novo protein truncating variants, intellectual disability, developmental delay, autism, unaffected individuals. And if it's a de novo protein truncating variant that happened in ExAC, so someone else has that exact same variant, or it disrupts a gene that has PLI less than 0.9, there is no significant difference in the rate between cases and controls. All of the significant difference falls in that tiny subset of de novo protein truncating variants that aren't seen in another individual and disrupt a highly loss of function intolerant gene. So this is how we've been able to use these two pieces of information to really highlight a subset of de novo protein truncating variants that appear to contain the signal. And again, we're jumping up now to odds ratios from 2 all the way up to 6.7. So the hope is, again, to be able to use some of the missense studies that we've started to do within the ExAC population to do a similar type of thing, but that is still in earlier days. So at that point, I've left plenty of time for questions. But big shout out to Mark Daly, who's my PI. So I can take that, or you guys can just go get more food if you want.